Good morning. Today in practical class, we will learn how to prepare a whole mount of the given material. There are different ways to make a microscopic preparation depending on the material and the type of study. To study the histological details of a tissue, we prepare thin sections. On the other hand, to study cell division like mitosis and meiosis, we squash the material. Here, we are preparing a whole mount, that is, mounting the entire material without squashing or sectioning. This is done to study the morphological features. We can prepare whole mounts of planktonic forms, larvae of helminthes, crustacean larvae, Obelia colony, fish scales, etc. The aim of this experiment is to prepare a whole mount of the given material. The material given is the scale of fish. Required materials: a fish, forceps, needle, and a few glassware like petri plates, cavity block, micro slides, cover slip. We need a spirit lamp, matchbox, and a dissection microscope. We also need a few chemicals like 10% potassium hydroxide, grades of alcohol, eosin stain, and glycerin. I have arranged the materials required for the experiment: a fish, 2 petri plates, a cavity block, forceps, needle, some clean micro slides, cover slips, a dissection microscope, spirit lamp, and matchbox. The chemicals we require are 10% potassium hydroxide, different grades of alcohols like 70%, 80%, 90%, 100%, eosin stain to stain the material, glycerin to mount the material, and some clean water. Before we take out the scales, take a petri plate and add 10% potassium hydroxide. Keep it aside. Hold the fish. With the help of forceps, slowly take out the scales. You can take 2 to 3 scales and transfer it into KOH. We have to heat the scales slightly so as to remove the mucus and the outer covering. So hold the petri plate and heat it just for 2 minutes or so. If you overheat, then the scales will start curling. After slightly heating, transfer the scales into another petri dish containing clean water. Wash the scales thoroughly in water so as to remove the traces of potassium hydroxide. Take out the scales and transfer them into a cavity block. We have to now process it through different grades of alcohol. Add a few drops of 70% alcohol. Cover the cavity block so as to prevent the evaporation of alcohol. After about 3 minutes, remove the cover and slightly tilt the cavity block so as to remove 70% alcohol. Now add a few drops of 80% alcohol. Cover the cavity block and leave it for 3 minutes. The process of keeping a material or a slide or a tissue through increasing grades of alcohol is called as upgrading. This is to remove excess water so that the material takes up the stain properly. Likewise, after 3 minutes, remove the 80% alcohol and then process the scale in 90% alcohol. After processing the slide in 90% alcohol for 3 minutes, remove the alcohol. We have to stain the material now. Add a few drops of eosin stain. You can use safranin also. Leave the scale in the stain for about 3 minutes. About 3 to 4 minutes later, remove the stain. Add 100% alcohol. Leave it for about 3 minutes. After about 3 to 4 minutes in 100% alcohol, remove the stained scales. Take the scale onto a clean glass slide. Add 2 drops of glycerin. Take a cover slip, hold it carefully and then slowly lower it with the help of a needle. Observe the scale under dissection microscope. I have kept it under 20x magnification. I have given the procedure here. After performing the experiment, you have to record your observation and give the result. Discussion. Fish have an exoskeleton of dermal scales. There are 4 main types of scales: placoid, ganoid, cycloid and ctenoid. Placoid scales are present in cartilaginous fish like sharks and rays, whereas ganoid scales are present in lower order bony fish like sturgeons and paddlefish. Cycloid scales are present in most of the bony fish like carps and herrings, whereas higher order bony fish like perch and sunfish have ctenoid scales. We observed a cycloid scale today. Cycloid scales are smooth-edged scales predominantly found in teleost or bony fish such as salmons and carps. These scales are circular in outline, thick in the center and thin towards the margins. They are flexible and translucent. Each scale is embedded in a small pocket of dermis and they overlap each other. This allows greater flexibility in movement. As a fish grows, its scales also will grow, adding concentric lines. These lines can be counted to estimate the age of a fish.